All right, folks, here we have it. Probably the most important lecture of this chapter, going about this topic of central dogma of biology, really, or molecular biology, and that is that DNA is used to code for proteins, and so now we're really going to dive deep into how this process works. We know the key players of the game, the RNAs. We know that DNA stores the genetic code or the genetic information, but really all of this is going to come together in this lecture where we look at how the key players in the game, mainly the RNA molecules, the messenger RNA, the transfer RNA, and the ribosome, which is made up of the ribosomal RNA, rRNA. We're going to see how all those come together to eventually make our amino acid sequence, which ends up being our protein after it's gone through all of the amino acid side chain interactions and the twisting, folding, and taking shape and such. So I love that the last chapter we had was about proteins, because remember, our whole goal is to make a protein, and so I love that you already have the protein chapter out of the way, because you know what we mean when we say that we're making an amino acid sequence. You already know what that amino acid sequence is eventually going to do. And so with your firm understanding of what a protein is, hopefully we can go through all of this pretty quickly, describing how, first of all, the process of transcription occurs, and then how the process of translation occurs. In between transcription and translation, we'll talk about genetic code, because it's very relevant to understanding how the transcript is translated. All right. So, but basically, protein synthesis can be broken down into two major phases: transcription and translation. Right? We need to transcribe some RNA, and then we need to translate that RNA and read it. So how does that work? So follow with me, if you will, into the lesson here. Basically, transcription is taking the information that's in the DNA and turning it into a transcript, which is going to be used to translate. Remember, we don't actually translate the DNA. The DNA stays in the nucleus of the cell. We don't mess with it. So what do we need? We need some information that is very similar to the DNA, and it's actually going to be an RNA transcript, which we call messenger RNA, which is gonna be that molecule. So here's how it works. Here's a good picture. I like pictures to teach from. So basically, you've got your DNA double helix, right? Well, and you got maybe some gene on the DNA that codes for a specific protein, right? Whatever this protein is going to end up dictating one of your traits, probably. So that's why we call it a gene. A gene is just a segment of DNA that ultimately is going to code for the assembly of one protein. And so now you can have, by the way, multi or polygenic traits, where the trait like eye color or hair color or skin color is actually polygenic, meaning it's actually going to be more than one gene that code for more than one protein that ultimately end up making your trait, your hair color or eye color. And so it's a little more complicated than just one gene makes one trait, but one gene means one protein. Okay, and so we have a start spot and a stop spot. Okay, we're going to start in one place and end in another. We're not going to read the whole DNA molecule. Remember, it doesn't code for just one protein. The DNA is super long. We're only looking at one section. And so if we're only going to be looking at one section, or the analogy I use in biology, because I teach this same stuff in freshman biology, is this is like a chapter. If the whole book is like the DNA, we just want to read one chapter at a time. And so we're going to read just this chapter. If we want to read that chapter, we obviously have to open the book. Okay, so you're going to have to open up the DNA molecule. Just like we did in DNA replication, you're going to have the same thing occur. You're going to have an enzyme called RNA polymerase. RNA polymerase, that's just going to be this brown blob here. It's an enzyme. It's basically a protein in and of itself. It's going to be a protein, or that acts as an enzyme that is able to open the DNA, unzip it. It basically unzips it. If you remember, these nucleotides, or these nitrogenous bases here, are held together by hydrogen bonds that are kind of weak. And so RNA polymerase has the ability to break those hydrogen bonds temporarily so that the template strand of the DNA can be interpreted, and then the DNA will come back together. But as it's being interpreted so that we make the newly made messenger RNA, it's using, you guessed it, base pairing rules. So if the RNA polymerase is, now it's kind of neat, the RNA polymerase has two jobs. One of the jobs is opening the DNA. The other job is to read the DNA. And if it's reading the DNA, what it's doing is, if it sees, let's say, cytosine, then it's going to use base pairing rules and put a guanine there. And then it's going to look at the next nucleotide. And if it sees adenine, it's going to put not thymine, but uracil, because remember, we're making an RNA transcript. And so messenger RNA is going to have uracil instead of thymine. So it would put a uracil. If it saw guanine, then it would put cytosine. So it basically just keeps reading the DNA template strand, keeps reading the template strand, and adding nitrogenous bases, or adding nucleotides one at a time, to form a newly made RNA that is not identical to the DNA template strand. But since we use base pairing rules to make the RNA, if we just use base pairing rules again to interpret the RNA, we'll be right back to the DNA essentially, wouldn't we? So as long as we use the same rules, we're good. We're okay. We won't mess up the genetic code, and I'll go over that in a minute. But for now, transcription simply refers to the process of, and I'm sorry, I have the direction kind of wrong here. Here's the direction of transcription. Transcription is simply the process of reading this template strand and producing a newly made RNA strand. At the end of the day, the DNA is going to go completely back to its reformed DNA helix, unscathed, like you won't even know you did anything to it. And then you're going to end up with some RNA, messenger RNA, which then can be translated. And so simple problems that the book has here for you is, like, let's say this is your DNA molecule, and that this is the template strand. Now, when I do this for biology students, I always put both strands, because DNA has two strands. I don't want to just put one strand because it's kind of misleading. You should have C, A, T, C, C, T, A, T, G on this strand at the bottom, going from 5' to 3' technically. And I'll probably do that on my test too, by the way, guys. So, but this is the strand we're concerned with because it's the template strand. You're going to use base pairing rules. And also, when you see C, you're going to put a G. When you see T, you're going to put an A. When you see A, you're going to put a U. That's the only difference with RNA, really, is that you have uracil instead of thymine. Now, you do have ribose instead of deoxyribose, but you don't need to show that here, right? So basically, make sure they're anti-parallel again. By the way, if the template strand goes from 3' to 5', your messenger RNA is going to go from 5' to 3'. It's anti-parallel. But they're complementary. Notice that we use that word a lot in genetics, in DNA and RNA and all that. This strand is complementary to this strand. It's not the same. It's not identical. It's not whatever. I heard a lot of funny words used. Complementary is the best word you can use. It's complementary. And so it's essentially the same as this in the fact that it carries the same message, but complementary is the better word. And so now that we have the mRNA sequence, we can do something with it. But here's some more problems to try on your own. And so real quick, though, I just wanted to point this out, that where we are in the scope of things, if this was the cell, if this white box here was a cell, then transcription took place in the nucleus. So this is the cell nucleus, all in blue. Remember, for eukaryotes at least, DNA is contained in a nucleus. For prokaryotes, there's no nucleus, but for eukaryotes, they do carry a nucleus. And so when the RNA strand is made, the messenger RNA is made, it then can go into the cytoplasm of the cell, which is the jelly-like fluid outside of the nucleus, but still inside of the cell membrane. Let's just say that this square is the cell membrane. Then it's in this cytoplasm where actually translation occurs. And so for us to take this mRNA and translate it, it first needs to come out here into this cytoplasm. So that's what you can see here. The RNA, and we're going to come back to this image too when we talk about translation. There's some really cool stuff I want to mention that's in this diagram. But I just wanted to point out for where we are right now in transcription, this took place in the nucleus. Okay, you can see here we looked at these bases, and we used complementary base pairing rules to produce this pre-mRNA. Let's talk about this just for a second, because I don't know if the book does or not, if or when it does. But before we make our actual mRNA strand, there's some pieces that get cut out. And then there's actually some pieces that stay in. Well, actually, there's obviously pieces that stay in because that's what you have here. And there's terms for that. The exons are going to be what's expressed. So everything that stays in are going to be little pieces called exons. They get spliced back together. All the little pieces that come out are going to be called introns. So it's a little bit of editing that's done first. And then the mRNA can leave the nucleus through what's called a nuclear pore. So this opening is called a nuclear pore. It's an opening in the cell nucleus that allows this messenger RNA to come out. All right, cool. So then it'll come to the ribosome and be translated. And as it's translated, whoa, what happened here? Oh. As it's translated, we're going to use genetic code. So the way genetic code works is there's going to be a sequence of 3 nucleotides, which we call a codon. A codon. There's going to be a sequence of 3 nucleotides found on the messenger RNA that we call a codon. And so once we've approached a codon, then we'll look at the next 3 nucleotides, and that'll be the next codon. And then we'll look at the next 3 nucleotides, and that'll be the next codon. And so a codon is just a sequence of 3 nucleotides in a row. Sometimes they're called triplet codons because they're just 3 nucleotides. Like, let's say you were looking at the messenger RNA and you saw UCA, that would be the codon. And that codon always codes for, at the end of the day, a specific amino acid, and we'll talk about how that all comes together. But as far as that codon always coding for this amino acid, that's true. Same thing for UGC, that always codes for cysteine. Okay, now remember we had 20 amino acids, and so here's the codon wheel you might remember from freshman biology. In our freshman biology book, you have this wheel. We use this, and I think we did some cool lessons where we took out the amino acids and we put letters there, and we made a little secret code in DNA. But basically the way you read this wheel is you start from the inside circle and you work your way out. So if you had a codon of A, U, G, you'd start here with A, then you'd find U. So where's U? It's right here. Then you'd find G, which is right here. So A, U, G, and that codes for methionine. That's actually the start codon, by the way. That's how RNA polymerase knows where to start forming the polypeptide. So methionine is your start codon. AUG is your start codon. Let's see. AAA would code for lysine. Notice something though, that AAG would also code for lysine. So AAA will never code for anything else but lysine. But it's not true to say that there are no other codons that code for lysine, because AAG does, right? And for some of them, like, check it out. If the codon is GG, if it starts with GG, it doesn't matter what the third nucleotide is, because it could be any 4 of these. It'll code for glycine. All right? And so you're probably asking yourself, okay, here's our codons and here's our amino acids. How do we start forming our chain of amino acids, and how are these codons interpreted? Like, that doesn't make any sense. And so let's start moving on to translation, because that's really going to make this whole picture come together. I just wanted to point out that in your book, they don't have a codon wheel. Instead, they have a genetic code table, I guess you'd call this. And this is actually more common and more professional, really. You should get used to using these. And it works the same way. Instead of starting into the inside of the circle and working your way out, you start on the left and you just work your way over. And then your amino acids are abbreviated here. So this is way more professional. You'll see this in most textbooks and stuff like that. So like, remember AUG was our start codon for methionine? Check it out. Here's A. Here's the first base, A. The second base, you're going to go here, 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 here. Remember, we're doing AUG, so it goes A. We're going to go U. Scroll down here. Now, what are we? AUU, AUC, AUA, or AUG? Well, it's AUG. And so if you don't want to look here, you can also look over here where it says third base. So it's G. So there, AUG codes for met or methionine. Okay. All right. So this is a total universal genetic code. So the messenger RNA in a potato is going to have codons that code for these same amino acids. So if you were to take genes from one organism and put them in another organism, you're going to get the same proteins being made, essentially, honestly. And so the body doesn't know the difference. When a ribosome goes to start translating messenger RNA, it's going to make, these are going to be the 20 amino acids and these are going to be the codons that code for those amino acids. It's totally universal. That's what's so awesome about life. Okay, so how do we translate them, though? How do we go from these codons to the amino acid? It's going to be the transfer RNA. So transfer RNAs are really neat molecules that have anticodons to the codon. And again, the anticodon, the relationship between an anticodon and a codon is just base pairing rules. Okay, I don't like to say opposite. It's complementary. So if it's UCA as a codon, it's going to be AGU because A is complementary to the U, G is complementary to the C, and U is complementary to the A. So the anticodon is going to complementary base pair with the codon, and as the tRNA does that, it's also going to bring an amino acid. In this case, it's going to bring serine because UCA codes for serine. Now, here's a common mistake. A lot of students will look at this anticodon and they will think that codes, they'll start using this to decode it, to figure out what amino acid, but it doesn't work that way. You're always going to look at the codon. In fact, this is a codon wheel, not an anticodon wheel. But it's tempting, and here's why. It's tempting to use the anticodon because look, let's go back to this picture now. I think this is going to be the best way to teach this. The transfer RNA has the anticodon and has the amino acid. So it's tempting to look at the anticodon, look at that codon wheel, and use this sequence to figure out this amino acid. It doesn't work that way. The information for the protein is found, well, essentially in the DNA, but then it's found in the mRNA transcript. Okay, the anticodon is just only there to recognize the codon. That's it. The anticodon is just simply for the transfer RNA to recognize the codon and be able to bring the amino acid. And this is actually how it works. So this is a great picture. So let's say this is our messenger RNA, which it is, right? It's gonna get interpreted at the ribosome. So ribosome here is pointing to this kind of like white structure, kind of a blobular structure, and it basically has a large subunit and a small subunit at the bottom. And those two pieces will come together, and in between the large subunit and the small subunit is where the messenger RNA gets fed through. Okay? And when this whole growing polypeptide chain is over with, this large subunit will disassociate from the smaller subunit once a stop codon is reached. So we'll go over that when we get to the end. But for now, I'm just trying to orient you guys with where we are and where everybody's at. This structure here is called the ribosome. And remember, technically, if you look a little deeper, the ribosome is made up of ribosomal RNA and proteins. All right? But really what I really want to show you here is the transfer RNA and how they're doing their job. As the messenger RNA is fed through the ribosome like a $1 bill in a vending machine, the transfer RNAs are coming and they're recognizing the messenger RNA by way of the anticodon complementary base pairing with the codons. Okay, AUG codes for methionine, doesn't it? Yeah, it codes for methionine. And so that's the codon for methionine. Well, it's going to be TAC as the anticodon for this codon. And so guess what? The transfer RNA that brings methionine is going to have TAC as its anticodon. And this would be methionine. I know that. It's not labeled, but I know that this is methionine. All right. Well, then then everything will get shifted over one. So you actually have 3 sites here. You have the entry site, and then I forgot the name of it, and then you have the exit site. So once the transfer RNA comes and it drops off its amino, so the transfer RNA comes, looks like it's going this direction. So the transfer RNA comes, it pops down here at the A site, the access site. The ribosome helps to weld, if you will, to weld this amino acid to this one. Now, you already remember that that's a peptide bond, right? That's a dehydration reaction. And so it's actually the ribosome that is able to do that. It's able to do the reaction that takes place to form this new peptide bond. And then everything will get shifted over one. And then you'll add another amino acid will come and drop its amino acid off. And so you eventually just form this long growing polypeptide chain. So the transfer RNAs that come into the A site, and then they go to this site, and then they go to the E site. When they leave, they no longer have their amino acid attached, right? Why is that? It's because it got attached to the polypeptide. So these transfer RNAs are only coming to drop off their amino acids. Then they're going to leave with no amino acid. We call that uncharged. So here's a charged transfer RNA. Here's an uncharged transfer RNA. Now you might be wondering, can this get used again? Sure. As you eat food and you eat amino acids, they will be recharged. And then your body will have them floating around in the cytoplasm just ready to go for this process. And it's so fascinating that your body is constantly assembling proteins all day long, and this is happening like in milliseconds. It's fascinating how fast this happens, and it takes so long just to lecture about it. And so at the end of the day, you're finally going to have a long polypeptide chain, which then eventually gets folded and twisted and taken whatever secondary, tertiary, and quaternary shape that the protein might take. But this chapter is essentially going over how we can transcribe messenger RNA, how we can translate it using genetic code at the ribosome using transfer RNA. And so to help with that too, I just wanted to show you kind of a quick animation that might make this a little easier to understand.
Here is a cell, the basic unit of...
You basically have, again, the cell that has all the normal components in the cytoplasm or whatever, but it's in the nucleus where the DNA is stored. And so remember, it's that DNA double helix that's going to contain the information of what proteins to make, which the DNA is usually all packed up into chromosomes, right, during certain times. And so it's important to unwind the DNA. Remember, this process was called DNA condensation. Whenever you wrap around 8 histones to make a nucleosome, and then you coil, and then supercoil, and then condense it even further, you can condense it into a chromosome. But if you want DNA double helix, you got to unwind it like this. And so usually DNA, if it's not in the chromosome form, it'll just be in stringy form like this called chromatin. And so you do have to separate it from the histones, but then you can interpret it. So you actually have to unwind it too. And believe it or not, if you want to get super technical, you actually have to straighten it out as well. So it's like a twisted ladder. You actually have to kind of straighten it out so it doesn't get all too tightly twisted. And so RNA polymerase anyways is going to be the enzyme that opens up the DNA helix. As it does that, it reads just the template strand down here. And as it's reading the template strand, it's adding nucleotides to a transcript one at a time using the base pairing rules. And then behind it, the DNA is getting put back together. And so here you go. Now you have your messenger RNA transcript. And again, we talked about some of the pieces will leave. There will be some editing that takes place. So there might be a section that gets edited out. You take that out and then splice it back together. Then that mRNA transcript will leave the nucleus through a nuclear pore, head out into the cytoplasm to find a ribosome, right? It's looking for that ribosome party flyer. And here it is, the large piece and the small piece of the ribosome. Remember, this is made up of rRNA and proteins. Here's the mRNA, and there's the tRNAs. The tRNAs have to be the coolest molecules. They're the ones carrying the amino acids. This is what we're doing this for, is to make amino acid chains. So transfer RNAs are huge. They're key. I'm going to pause it here because check it out. Look how they work. They have the anticodon, but look how they also have the amino acid, and look how the single-stranded nucleic acid coils in on itself, and the nucleotides will like base pair with other nucleotides on the same transfer RNA molecule. Isn't that kind of cool? And so they end up usually having this cross or T shape. So there you go. There's your transfer RNA. Again, it will recognize the codons by base pairing rules. Okay, if we pause it here, you see AUC is the complementary base pairs with UAG. And as everything's getting shifted over, amino acids are being dropped off one at a time, and you're forming a growing polypeptide chain. This is called elongation. And there you go, see? And now eventually, the cell is going to reach a stop codon, or the mRNA is going to have a stop codon on it, which it doesn't show there, and there won't be an amino acid on that transfer RNA. And then remember, the amino acid gets folded, twisted. The amino acid chain gets folded or twisted into protein shape. So if you want to learn how to sequence the human genome, watch that video. I'm not doing that right now. So where were we? Yeah, in the book, they basically just show you these little boxes. To me, that didn't make a whole lot of sense. So that video I found was super helpful. Now, the one thing that video did not go into, though, is these 3 phases of translation. So when you're translating the messenger RNA transcript at the ribosome, we have a phase called the initiation phase, the elongation phase, and the termination phase. And so here's how it works. It's not that complicated. Basically, initiation is when the first codon is reached, and it's always AUG. AUG codes for methionine, and that's your start codon, essentially. We don't call it a start codon, but AUG is, we don't call this the starting amino acid, but AUG is the start codon. Okay, that's how we know how to start. So there would be initiation. You have your first transfer RNA coming with your first amino acid. Elongation happens when a second tRNA will bind. Now you can start elongating the amino acid chain, the polypeptide chain. So you make a new peptide bond there, which you remember from the last chapter what a peptide bond is. Okay, and then this transfer RNA will leave with nothing on it, because remember, it dropped off the amino acid. A new transfer RNA will come and recognize that codon by its anticodon and drop off, what is that, glutamic acid maybe, and then that'll attach, and then you'll just have a huge long growing polypeptide until finally you reach a stop codon here, at which point the transfer RNA will not have an amino acid. Instead, it'll have some other enzyme or some other molecule that helps this process stop. The large ribosomal subunit and the small ribosomal subunit will dissociate, and the polypeptide then will be carried out to be taken to the Golgi apparatus. Actually, the Golgi apparatus and the endoplasmic reticulum will do protein modification. So that's actually where all that twisting and folding and this stuff takes place. The Golgi apparatus is like the post office for proteins, and so this will show up to the Golgi apparatus inside of a vacuole. The vacuole will merge with it'll fuse with the membrane of the Golgi apparatus, and then once this is in the Golgi apparatus, it'll get sorted out, folded, twisted, and then it'll get packaged and put into a new vacuole and either sent out of the cell or kept inside of the cell wherever the protein is needed. But termination, again, translation continues until a stop codon is reached. There is no transfer RNA that contains an anticodon, complementary to any of these stop codons, so that's actually why. That's actually why it stops, because there's no such thing as a transfer RNA that has an anticodon for those stop codons. Excuse me, I had that wrong. I'm sorry. And so protein synthesis ends, and the protein is released from the ribosome. Often the first amino acid in the chain is methionine, though, but sometimes it's not needed in the final protein, and so it is removed after protein synthesis is complete. So if you were going back to the protein chapter and wondering why some of the proteins didn't have methionine as the beginning, it's probably because it got removed. But it is needed to start the process of translation, though. So figure 17.10 shows a representative segment of DNA. This would be a DNA segment. It also shows the mRNA segments with the codons that complementary base pair. It also shows the tRNA anticodons that base pair with the codons, and then it shows the polypeptides that correlate with not the anticodons, but correlate with the codons. It's real important to remember that when you're looking at this codon wheel, that you're using the 3-letter codons on the messenger RNA to figure out what amino acids there are. And if you're curious about the stop codons, there's only 3 of them. There's AUA, AUG, and UG. I'm sorry. UAA, UAG, and UGA, the code for stop. In other words, there's just no amino acid that translates to, and so there's no transfer RNA either. All right, but around the rest of the wheel, here's something to note, though. You might wonder, why is there more than 20 amino acids here? It's because arginine shows up here, but also there. Serine shows up here, but also there. It's because there's different codons that code for serine. It could be AGC, AGU, or it could be UCU, UCC, UCA, UC anything, right? All right. So basically, then we can talk about mutations and what happens if the sequence gets messed up and all that. We can do that next time. But for now, I know that was a lot, but isn't that kind of spectacular that the DNA, the sequence of nucleotides in DNA, ultimately, after transcription and translation, that sequence will determine what protein is made via that process you just saw. And so it's very important for a cell to contain a ribosome and transfer RNA to be able to do that. So here's what makes, if you're still doing your project, by the way, on the coronavirus, here's what makes a virus kind of interesting. Viruses contain not only a phospholipid bilayer membrane like most cells do, they even contain surface proteins that are used for like dog tags or name badges that, they're used for different reasons. In the case of the coronavirus, it's to attach to an ACE2 inhibitor receptor site on your lung cell, which will allow it to be taken in. And so, but it also has DNA inside of it. Actually, coronavirus is an RNA virus. So it has RNA, it has messenger RNA already in it, ready to go, ready to be translated. But it has no ribosome. So what does it do? It uses those surface proteins to attach to your ACE2 inhibitors on your lung cells, and then your lung cells will ingest the coronavirus, and then it'll translate the RNA inside of the coronavirus. And what kind of proteins do you think it's going to make? It's going to make those surface proteins that are on the outside of the coronavirus that you've been seeing pictures of. That's what makes this coronavirus deadly, because those surface proteins are a whole lot different than the coronavirus's proteins we've been used to, because the common cold is essentially a coronavirus. But the new surface proteins, those spike proteins, they've changed through whatever reason. There's lots of theories out there, but theories are that it went from a bat to a pangolin and whatever inside of an animal market, and then allowed it to jump species. And so now we've got this thing that those surface proteins, we don't have any antibodies for those, and so yet at least, and so they. But the point I'm wanting to make right now is that the virus does not have a ribosome, and so it has to use your ribosomes to interpret or translate the RNA to make those proteins. Otherwise, the virus is actually harmless. If you can find a way and there is a way, there's a drug out there called remdesivir. I encourage one of you, please, to do your project on that, because I'm quite fascinated with it. Remdesivir is a nucleotide analog, which acts like one of the nucleotides. It looks really close, but it's not the real deal. So it actually ends up messing up the process, and so the RNA won't get translated. You won't make the proteins, and so even though you get the virus, it will essentially be harmless to you because your body won't keep replicating, or it won't keep translating the RNA, which will eventually mean the virus won't get replicated. So yeah, there you go. Transcription, translation. And again, this whole process going from DNA to a protein is called the central dogma. There's actually one thing I knew I left with you as you went off to college in either biology or biochemistry, that it would be that. That's why I focus on it in freshman biology, and I brought it up in this course so much, because students actually get kind of lost in this process, and but it's also a field that is wide open for study, so it's very popular in schools right now, and you really need to understand this process if you want to sound intelligent in college, because it's going to be one of the major topics on SAT tests, you name it. So hopefully that was helpful. There's a lot of videos out there about transcription and translation if you want to watch them. There's lots of images on Google that might help you. Anyways, blessings.